research this, but basically
I'm asking incredibly negative consequences.
Organisms are better chemists than us. I would like to know for a question, but if you need, that's all. First, I want to talk a little bit just so you guys know who I am, because it's related to me. The rest of you probably don't, because I graduated about 3 years ago, I think in the class of 2020 at UC Berkeley. I've spent working in the Renata lab at Scripps, and now I've been in the chain group at UC Berkeley for about a year and a half now. I want to give you guys a little bit of background on the sort of work that my lab does I've been working on. What is a product? I'm going to actually take suggestions from the audience. What do you think one of those is? Someone have a volunteer? Okay, that's a good suggestion. Any other ones? Okay, so things that come, think asparagus, CL. So maybe they come from nature. And that is correct in some sense. I like specific molecules like paclitaxel. These are what are referred to as secondary metabolites. They're not necessarily as part of living, but as to do something that's evolutionary. For example, comes from a bacteria known as in disease, and paclitaxel comes from bacteria. One either different, but also and then is the bacteria as part of redox warfare. These also have different uses in humans, right? Piocenonin is actually implicated in disease. Paclitaxel is an anti-cancer drug, right? So we can see there's a lot of diversity in both the right? So what is this? So this is an amino acid, but it's not one of the ones that you probably know. You know when people talk about, oh, I need more protein or whatever. Proteins, basically. But that's a very limited set of things. We can do a lot. 20 amino that do things in our body, but they only do some things, right? We can set the 20. And these have very diverse things. Ornithine, for example, is part of metabolism. Is a bio. So these are all really diverse, as we can see. The canonical and non-canonical have a much larger range of diversity than canonical. And that's because there can be effectively infinite of them. Any modification will make non-canonical. These non-canonical amino acids, which is the like T5 highlight, is to make complicated molecule. 3, you 2? Okay, so that's the trees take 19. How many do you think it took humans to do when the first time they made this?
27.
You're being too nice to humans.
46.
Yeah. If you are good at math, you know that 46 is in fact bigger than 19. So, as I've said, the tree is making these enzymatically, but like, what are enzymes? Enzymes are things that are proteins that can modify substrates, which are things which are molecules that they take in, modify, and then they can push out. So I'm going to remind you guys of the central dogma for biology, because we don't care about the biological processes in the and use those genes, and a bunch of biology happens, and they make enzymes for us, which we can use. That's all we care about. So why do we care about these natural products that we're talking a lot about? So 80 new drugs and natural products. Rationale for why we want drugs to treat novel diseases or diseases that were previously untreatable. And how do you find this? So this is, I'd like to emphasize, one of the ways you can find products. It's a technique called genome mining, where you have a collection of bacterial strains and you take all the DNA to see what's the order of all the DNA in there, and then we can look through all that DNA and find these things, gene clusters, units of the bacteria, enzymes. As I said, that. So, background on where my lab sits in the world. I want to give you guys a little introduction to the previous work my lab has done before delving in. So, previous work by a previous graduate discovered this gene. So the things that I want to note are best D, best C, and best B. And so that thing on the left, or right, I guess, originates it and then makes it all the way in the end to this terminal amino acid. That's something I want to narrow it. So this, as we can see, is lysine, but with all the explicit. Select exercise in this process, right? Is we see we put in a halogen, but by the end it's gone. Intermediates so that it can do further reactions and then finally conclude with a product that might not have that halogen. That's really interesting because we can find these new natural products like this, but for this halogenation step sometimes to find these non-halogenated. So family of them. So these are related enzymes that will take different substrates. Be halogenated from position and eat. It's able to halogenate them in different places times. And so this really tie into what I'm talking about here with all of making these and finding these new brings us to the sort of work I'm working on. To use these kinases, we can look for genes that are near them. Out, which kind of sucks. It leads us to these very. So we found which was it has these 5 genes, right? Later, we don't really care about that. It just because really know what that acid trend. We also don't really care. These are part of like the plants. That that we refer to as the PAZ cluster because it's from P pseudoformans, so PAZ. DNA that you can put into bacteria, and then they will make on them. And so when we it did two things. Originated because, and obviously this is a and this is in fact are not also that common in nature. So that's a pretty interesting result. We dubbed this concept, this compound pazamine because it's made by P azotoform. Because these genes in a bacteria, we were able to find out that this, what we thought was a serine hydroxymethyltransferase cyclopropanase. But what else can we do with this? We wanted to halogen as B. We wanted to see put if we feed amino acids, we get sizes of amino acids. So we did that. We proposed that, kind of cool. We can make so we want we're able to successfully get D to work. Which all very interesting. Use this halogenase mining strategy where we have this Hal family, and we were able to find a gene cluster from Pseudomonas azotoformans to find new non-canonical amino acids and use those new enzymes to do new chemistry. So, in conclusion, why are the enzymes better? Right? They're able to make like paclitaxel or just even compounds that more efficiently and better. I would like to acknowledge the lab, Professor Michelle Chang especially, and Max Sosa, who I've been working with, and Dr. Jeff Pelton at B3 NMR for all his help with 2D NMRs.